Hello, everyone. I'm Jan Reichert, Chief Operating Officer of the Antibody Society, and today I'm going to tell you about trends in the early stage clinical development of antibody therapeutics. I'll start with the basics, definitions, sources and limitations of the data, and our objectives in collecting this kind of data. I'll then discuss trends in the entry of these molecules into clinical studies. The focus of the presentation will be on that early stage pipeline. I'll do compare contrast of the status of the antibodies for cancer versus non-cancer indications. Then I'll discuss the format and targets specifically for the antibodies for cancer. The next topic will be trends in the commercial antibody therapeutics pipeline, specifically compare contrast of the early versus the late stage pipeline. The final topic will be trends in first approvals of antibody therapeutics, looking at the combination of the US or the EU compared to the rest of the world. I want to discuss the definitions briefly because that gives me an opportunity to tell you about our inclusion-exclusion criteria. Specifically to us, an antibody therapeutic is a recombinant protein-based molecule. It will have at least one antigen binding site that is derived from an antibody gene, and it will have been or is being evaluated as a therapeutic. So specifically, that does exclude certain things. It excludes polyclonal antibodies that are from a natural source. It excludes DNA, even if that DNA encodes an antibody. It excludes the Fc. It also excludes Fc fusion proteins. And of course, it excludes anything that is a diagnostic. We are looking at commercial development. So this is development by public or private for-profit entities. This specifically excludes then programs that are developed solely by nonprofit or government entities. For the clinical status, I will be talking about clinical status, and what that means is the most advanced clinical study. This excludes any early stage studies for molecules that have proceeded past phase 2, and that would include pivotal phase 2. So we're excluding any data for molecules that are pivotal phase 2, phase 2-3, phase 3 studies, or that have progressed into regulatory review or been approved. We are interested in innovation. Therefore, we're including molecules that have unique composition of matter compared to everything else. The data that we have does exclude biosimilars. First, that one should be easy. First is first. So it excludes any second or third incidents. And specifically then, the first in human studies is a singular event that occurs only once. A first global approval is a singular event that occurs only once. Antibodies to watch, I'll mention that multiple times. That phrase refers to an article series that has been published since 2010, and it does document the late stage commercial clinical pipeline. We rely on the company as, the primary source of information. We look at all of their public disclosures, press releases, presentations, meeting abstracts, any kinds of reports that they put out. We also look at information they put in the clinical trials registries. We do consider those trusted sources of information. We, of course, also include the regulatory agencies and WHO, the World Health Organization, as trusted sources of information. We cannot and we do not rely on secondary sources for information. And specifically, those secondary sources would include commercial databases. We do not rely on them. And that's really because our inclusion-exclusion criteria is very specific. As I described to you, we are looking for a particular subset of antibodies that are in development. And the commercial databases just simply do not have the filters that enable us to access that data. So we built our own little data set. We do find, though, that the commercial databases often lag in the updates, especially for terminations. And of course, there can be errors that occur during data processing. No matter who is collecting the data, this kind of data does have some limitations that I want to mention to you. There can be substantial lags between when an event occurred and when the company decides to say something about it. And of course, there can also be a lag between when the company says something, releases a press release or a cryptic mention in an annual report. There can be a lag between when that happens and we notice it. Particularly for those early stage molecules, companies may not divulge all the information. They may not even divulge that it's an antibody. So we do have to patiently wait and collect data and determine first whether it's an antibody, and that puts it in the data set to begin with, and then also to collect additional information as it's released, for example, on the isotype or on the target, even on the therapeutic area, because the therapeutic area may not be divulged immediately. Those phase 1 studies may be done in healthy volunteers. We usually can find an initiation date for clinical study. The termination date may be impossible to find. The company may just simply allow a molecule to disappear out of their pipeline without comment. In that case, there's really no way to say exactly when that molecule or when that program was terminated. The status can be inconclusive. ClinicalTrials.gov records are not always updated in a timely fashion, and the company pipelines are also not always updated in a timely fashion. We do find also that clinical phases are more frequently being blended. So I like now to refer to early phase versus late stage clinical studies, the early phase including phase 1, phase 1/2, and phase 2, late stage being pivotal phase 2, phase 2/3, and phase 3. Specifically for antibodies for cancer, we do find that a first-in-human studies is very frequently a phase 1/2 study. Now, collecting all of this information is a substantial amount of work, I have to say, but we do have specific reasons for doing it. It allows us to determine trends in antibody therapeutics development and thus be able to give you the presentation that I'm giving today. So that can be overall trends, or it could be trends in particular therapeutic areas, trends relating to specific formats or to specific targets or types of targets like the immune checkpoint modulators. It also allows us to determine clinical success rates for antibody therapeutics. As it's conducted by the biopharmaceutical industry in particular, then we can calculate the rates for transitions between different phases of clinical studies as well as the overall marketing approval rates. It does allow us also to assess innovation in the biopharmaceutical industry. Is the industry developing new and innovative molecules for patients who are in need? Now I'll proceed into the data-heavy section of this presentation. This graph illustrates the increase in the number of antibodies entering the first in clinical studies. The green bars show the number of molecules that entered clinical studies in any given year. The red bars are those molecules that are for cancer, with the blue bars being those molecules that are for non-cancer indications. The dotted lines are the 2-year moving averages. As you can see, between 2010 and 2014, those lines are relatively flat. The largest number that entered clinical studies in any of those years was 84. And the number that were for cancer versus non-cancer indications were reasonably similar. That changed in 2015. If you look at the dotted green line, you see that really increasing at a pretty rapid clip, especially relatively recently, increasing from 121 in 2015 to 286 in 2022. You also see a rather large gap developing between the dotted red lines and the dotted blue lines indicating substantially more antibodies for cancer are entering clinical studies relatively recently. Now, I will focus in on that early stage clinical pipeline, again, emphasizing that this is commercial clinical development. Also, I'll emphasize this is a preliminary analysis. We collected a lot of data. There's a lot of different ways of looking at it. So what I'll tell you about is our first attempt to describe this, starting with the status. The large pie chart on your left is the status for all of those molecules. And you'll see that there's a lot. The N here is over 1,000 molecules, with the most advanced phase of 56% of those being phase 1. About roughly 20% each are in phase 1, 2, and phase 2. But you do see a difference if you look at the cancer versus non-cancer indications. Those are the smaller pie charts that are on your right. The top one is the pie chart showing status for cancer, but you also notice that there's a lot more molecules. Over 700 antibodies are for cancer that are in this early stage pipeline. Non-cancer indications is 321. For cancer, the most advanced phase is 59% are at phase 1. You see at phase 1, 2, there's 25% with 13% at phase 2. For the non-cancer indications, phase 1, it's 52%. Only 8% of the antibodies for non-cancer indications are at phase 1, 2. At phase 2, you see 38%. So it's a bit of a difference there compared to the non-cancer, compared to the cancer indications. For the cancer only, those do tend to go into a phase 1, 2 more frequently as a first in human studies. So you see more molecules at phase 1, 2. For the non-cancer indications, they are slightly more advanced. They've been in clinical studies for a longer period of time on average, so you see more molecules at phase 2. In looking at format, there are a lot of different formats for antibodies. To keep this analysis reasonably straightforward, we decided to bin them in only 3 ways. We took all the formats that give you a monospecific antibody, and we binned those. Then the second category are all formats that give you a multi-specific antibody. The third format that we put together, those are the molecules that are immunoconjugates. So we'll look first at the targets for those monospecific antibodies. And that total was 436 of them. About 2/3 are canonical antibodies, so standard antibodies. About 1/3 are ADCs, and with a relatively small percentage of them radio-labeled. If we look at the targets, we did a frequency analysis to look at the top 20 targets, and I will not go through all of them. The slides will be available to you. I'll just touch on the top 3 targets here. They were Claudin 18.2, HER2, and PD-1. Looking separately at the antibodies for the canonical antibodies, that is the bar chart that is on your left. The most frequent targets were PD-1, TIGIT, and Claudin 18.2. Then you see CD73 coming in, as well as an array of checkpoint modulators. For the ADCs, you see a slightly different picture here, and that is the bar chart on your right. The most popular targets were HER2, Claudin 18.2, and TROP2. For these monospecifics, again, I wanted to mention we do look at innovation. So we were happy to see that 18% of the monospecifics do target an antigen that is unique to all others that are in this early stage pipeline, with about 2/3 of those targeting tumor, about 1/3 that are immunomodulatory, and 3% that target an antigen that's relevant in angiogenesis. For the multispecifics, this becomes rather complicated. So I will walk you through all of the graphs that are shown on this slide, starting with the center. In the center, you see a pie chart that is for all the multispecific targets. About 90% of them, though, are bispecifics. So I'll immediately turn your attention to the pie chart that is on your left, which is for the bispecifics that are not ADCs. For those bispecifics, about 2/3 are cell engagers, with 15% targeting tumor, 14% targeting immune cells. The most popular targets are shown in the bar chart in the lower left. Most popular combinations were CD20, CD3, PD-L1, 4-1BB, BCMA, CD3. So those cell engagers were really quite popular amongst the bispecifics. Now I'll direct your attention to the far right-hand side, the pie chart that really is just one color. These are the ADCs, they all target tumor. Most popular target very definitely is HER2, so that is the bar chart on the far right-hand side. And as I mentioned, like the monospecifics, the ADCs that are multispecific do have a tendency to target HER2. That is the most popular target. In looking at antibodies that target more than 2 antigens, so that is the pie chart that's on the top. Of those, some 83% are cell engagers. So again, that is a very important mechanism for these multispecific antibodies. In looking at the targets for all of those multispecifics, those are shown in the bar chart in the middle on the bottom. And you can see there is no popular target. All of the combinations of antigens are different for the multispecifics. So again, in looking at innovation, there are a lot of different combinations, and some 43% of those combinations were different compared to anything else in the early stage pipeline, with about 2/3 of them, though, being cell engagers, 20% targeting tumor, 10% targeting immune cells. Since the cell engagers were so important, we decided to look at a frequency analysis of the antigens that were on the immune cell versus antigens on the tumor. Not surprisingly, the most popular target for the antibodies that are the cell engagers, the most popular target on the immune cell side was CD3 on T cells. Most popular targets on the tumor side were PD-L1, BCMA, and CD20. We've seen those before. For the immunoconjugates, there were a variety of types, but the majority of them fell within 3 different categories: immunocytokines, bispecific immunoconjugates, and immunotoxins. So I give you some examples of those on this slide. For the immunocytokines, so this is the series of charts shown on your far left. Most popular target was PD-1. The most popular fused cytokine was IL-15, followed by IL-2 variant. So those two were quite common. For the bispecific immunoconjugates, the most popular target was PD-L1. The most popular fused protein, without a doubt, was TGF-beta R2. For the immunotoxins, there really was not a most popular target. They were all different. The most popular fused protein was Shiga-like toxin A. Now I will transition into looking at that overall clinical pipeline, just really emphasizing the increase that has occurred over the past 4 years or so. When I was writing Antibodies to Watch in 2019, we were collecting and analyzing data in November of 2018. That data is shown in the dark green bars here. I did the same type of analysis in November of 2022 in preparation for publishing Antibodies to Watch in 2023. And you can see that there was a really large increase. The percentage increase between 2018 and 2022 was 110%. But we were very pleased to see that that increase did occur across the board, both at phase 1, phase 2, as well as phase 3. So that really illustrates that the molecules are progressing very nicely through the pipeline. That Antibodies to Watch article series is really excellent documentation for what's going on in that late stage pipeline. And again, this is commercial development. If you look at Antibodies to Watch in 2010, published in mAbs, you'll see that 26 antibodies were documented in that paper. So if you go to the paper, you can find the identities of those molecules. By 2016, that had doubled, but it stayed rather flat. So through 2018, we were still at just a bit over 50 antibodies. So it's essentially doubling what had occurred in the 2010 article. So that's an 8-year span. In a 4-year span, it doubled again to over 100 in Antibodies to Watch in 2022. If you look at Antibodies to Watch in 2023, you'll find in the supplemental material the identities of 138 molecules. And we do expect the number to be larger in Antibodies to Watch in 2024. So again, that article series really is a very good source of information for that late stage pipeline, and it very definitely has documented this massive increase in the late stage pipeline. Well, that increase in the late stage pipeline has allowed an increase in the number of approvals. This graph shows the annual first approvals. So this is the first global approval, regardless of the country. The green bars show the total number of first approvals that occurred in any given year, with the purple bars being a first approval that occurred in either the US or the EU. The gold bars shows those approvals that occurred in any other country. And first, you can see, just looking at those green bars, or the 2-year moving average, which are those dotted lines, that there has been a very nice increase from just 1 approval in 2010 through to 20 approvals in 2022. But also here, I want to emphasize that the approvals that occurred outside the US or the EU have increased recently. The phenomena has always existed. There have been an occasional antibody here or there that was approved in a region outside of the US or the EU or a country, but it was relatively rare. Typically, the US or the EU was considered the first launch area, either the US or the EU. However, that has certainly changed, and that's represented by that closing of the gap between the purple dotted line and that gold dotted line. It is a recent trend, so we will definitely be keeping an eye on that and reporting on it in the future. With that, that is the end of the data that I have to present, and I just want to wrap up with just some key messages, which I hope were very clear by the data that I showed. In particular, that antibody therapeutics are entering clinical studies in increasing numbers, such that it's becoming hard to keep track of them all, particularly those antibodies for cancer indications. For the early stage clinical pipeline, the majority are monospecific, but about 1/3 are multi-specific. We do see that nearly 20% of the monospecifics and over 40% of the multi-specifics do target a unique antigen or combination of antigens compared to other things in that early stage pipeline. And that says to us that the biopharmaceutical industry really is engaging in research and development that is innovative and is directed towards addressing the substantial medical needs specifically for new cancer treatments. I also hope I've managed to convince you, based on the data, that antibody therapeutics are certainly entering the late stage pipeline in increasing numbers and also gaining approvals in increasing numbers. With that, I want to thank my colleague, Sylvia Criscioli, for her work on the analysis of that early stage pipeline and preparation of all those lovely figures that I showed you. And also, I want to thank the Antibody Society and, of course, their corporate sponsors. The Society does have a whole array of corporate sponsors, over 50 at this point. So I want to thank each of them individually by including their logos on these slides. And again, we would not be able to do what we do without their very kind support. So I thank them. I also thank you for your attention. Of course, available anytime to answer questions, and I invite you to visit our web resources page to find more information.